Okay, so sorry. I didn't mean to do that. Okay, so tomorrow's quiz for microbiology for 221. Why is it so blurry? For 221 is lecture quiz 5, and it will be on chapters 16 and 17, and it will be multiple choice only. Let's see, maybe I can make my, yeah, touch up my appearance. What's with that? Make this better. I guess that's it. Just get blurry me. It will be all multiple choice. No short answer. No, just multiple choice. Maybe some matching, and of course the multiple choice of which of the sort of like a true false, which of the following is not, or which of the following is a characteristic of whatever, blah blah blah. For 16 and 17, I did post a rather extensive PowerPoint for 16 that I did edit and put things in that I would have told you. I do not think I did not post a, yes, I did not post I didn't post a note outline for 16 because the PowerPoint was pretty in depth. But for 17, I believe there is a note outline, and highlighted on there are the figures. I updated them for the figures for the 6th edition. So those are the figures that we would have gone through for chapter 17 for diagnosing infections. Right. So I think that's about all I know for those things. I'm probably going to, yes, there'll be some matching, and the matching will be in short sets, 5 or 6 terms with 5 or 6 definitions picked from. Usually if it's longer than that, students can't see all of the terms or all of the, like if the definition is really long and takes up 2, I don't know why it's so blurry, takes up 2 lines, they can't see all of it at the same time in the box on Canvas. So it will be short. They'll be in, like I said, 5 or 6 little sets of matching. How is lab going for you guys? No one want, yes, no? Pretty good. Oh yeah, Zoe, I think I just replied to your email not that long ago. Anytime that you feel like you have to wait to get the bacteria to grow, that's perfectly okay. For the bread to mold and for the bacteria to grow, put them in a warm place, guys, put them in a warm place. And keep them wet, right? And just use tap water because that'll have enough bacteria in it to help with the mold growth. So I would keep them in a warm place. And if you're still waiting on that to mold, I would just get started on the actual bacteria labs, because those are going to take about, well, for me, they took like 9 or 10 days pretty much. So, but I didn't check this morning to see if my bacteria were growing. Buy a lot of bread and molds. Yeah, it's like a watched pot never boils, right? So a watched loaf of bread never molds. If you have any yeast, you could sprinkle it on there to help encourage. You get some yeast molds. Oh, good. Starting to look yellow in some spots. Yeah, so you'll get mold soon. Yeah, same. Seems like my bread's always moldy. Well, in the refrigerator anyway, but yeah. So yeah, hopefully you'll get some bread mold soon. I think I've mentioned this before. The microscopy portion is better with the bread mold, the brine shrimp, the wool that you have to look at, the frog blood, and I forgot the third slide that was in there. Looking at the bacteria under the microscope is not very exciting, unfortunately. And I personally, and I think I sent this in the email privately to Zoe, I personally was trying to hurry my bacteria along the slides to dry because I was trying to do it sequentially to see how long it would take you. So I was holding mine over the flame like they show in the one video. Then I was holding them over I have a space heater up in that room and I had them near the space heater. And I think that I fried the bacteria because when I looked at them under the microscope, they looked like they had been exploded. And that usually means there's still water in them. And then when you heat them up, they explode really fast. So my advice on that is two things. Skip the water completely. Just skip the water. Just take from your broth culture with your loop and put like 2 or 3 loopfuls on the slide. And I usually say, like when we're in real lab, and I think in one of the videos I said don't tap, because when you tap, the bacteria kind of aerosolize. You're going to have to tap because these plastic loops, they don't let go of the bacteria like the metal ones do. The metal ones, when you just lay them on the glass slide, the little loop full of bacteria just like disseminates right onto the glass. I found that didn't work so well for me either. I couldn't get my staph to grow on my Petri dish for the last exercise, and I think it was because I just tapped my loop and I didn't like smack it. So kind of look away so you don't inhale the bacteria, but I would gently tap my loop and I would probably do like 3 loopfuls of bacteria from the broth onto the slide and just completely skip the water. You'll get more bacteria, it will look cloudy, you'll get a really good smear, and then it'll probably stain better. I think I had mentioned that in the one video that I was going to try doing that, and I still haven't. I still have my old slides up there. I haven't stained my second set. So what you might want to try is follow the directions for your first set for slides 1 and 2 for gram staining. And then for your second set, just do loops full of broth, maybe. I don't know. And then stain them all at the same time. I mean, it's an experiment, you know, just the best you can. So that would be my advice on the staining. Sadly, they didn't, like I said, they didn't turn out that great. I think it's mostly because of the microscope. We really need 1000x magnification, and we really need immersion oil to actually see bacteria. So don't despair, just draw what you see. And then I think the questions that were in the virtual gram staining lab, those are really good questions. So I think you guys can go back in and review those for practice. And if you can't, somebody let me know, because I will put those back. I will put those on the lab report. I'm kind of waiting. I wanted to ask for your help for that today so that I know whether or not I need to put those questions on the lab report. So if you can go back in, if you wouldn't mind, somebody, and then shoot me an email this afternoon, if you can go back in and do those gram stain questions, because they were really good critical thinking questions, I thought. So I'm going to put just some generic gram stain questions on the lab report, like what the order of the stains was, what color you think they would be at step 1, step 2, step 3, step 4, and the questions that we normally have in the lab book. So I am going to put those in for the simple stain and for the gram stain. And then the last thing I have to add on there is some questions for the biochemical test. And because I haven't done them yet, and I don't know what you're going to get for your results, that's partly why I'm waiting. So I'll do those today, and then I'll know what kind of results you get for oxidase, catalase, and the TSI tubes. And then I'll ask you appropriate questions based on what the results I think you're going to get, rather than just giving you questions that you might not be able to answer because your results may not turn out that way. So that's kind of where we stand with the lab report. I'm hopeful that I will finish that up today or tomorrow, get that posted for you. So it's a fill-in, the lab report, and then upload photo. It would be most helpful to me if you could sort of upload 1 photo of you with all your results, so I don't have to look at 6 photos for every person, or 4 photos for every person. Yeah, the idea is to see that you actually did the labs. Apparently in Bio 1, it's pretty rampant that people are paying other people to do their labs for them. I should be doing that, right? Instead of this, it's people are getting like $50 a lab if you do someone's lab for them. And there were several Bio 1 kits that no one purchased, even though we only bought enough kits for the number of students, and there's more kits left than people who've withdrawn. So I think some of the people who never bought the lab kits are instead just paying people to do their experiments. Okay, so don't do that. And if you need help with your results or you want to talk to me about your results, definitely let me know. You can still view the labs. Okay, thank you, Whitney, for checking on that for me. So that's awesome, right? So go back and look at the questions for the Gram stain lab before and the pictures, because the pictures in the virtual lab are way better than the pictures you're going to get. Not to be discouraging, but so I don't care if you draw the pictures from the virtual lab on your lab report to me, okay? Because it's more important to me that you see what sort of should have been the results, but the going through the staining procedure was important to us, which is why we purchased that part of the kit. We wanted you to practice going through the steps of actually staining something. So yeah, that didn't turn out like we thought, but whatever, best we can. Yes, dear, I can't hear you. No, yes. All right, I'm gonna stop.